Okay, good deal. So, how many people do I have from computer science in the room? Almost all. All right, anybody biology? Okay, good. That'll focus my talk today, actually. This is kind of inspired by Richard Feynman, who's an amazing physicist. I don't know. I wish everybody would watch all of Richard Feynman's videos online. They're wonderful. He was a physicist who worked at Los Alamos and built the first nuclear bomb, and he invented Feynman diagrams. It's amazing. He's an amazing scientist. But he wrote a paper in 1959 called "There's Plenty of Room at the Bottom." And I'm just running a little animation here that shows, like, this is actually called scale. It's just zooming in on the universe, and you could see the relative size of particles and things. But he said back then that he thinks that a $1,000 prize for the first guy who makes an operating electric motor, a rotating electric motor which can be controlled from outside at 1/64th inch, which should be done. And oddly enough, a couple years later, he had to pay the $1,000 for that small little motor. In 1947, this is the very first transistor that was built. It's a picture of the very first transistor, solid-state transistor. And that kind of ushered in the era of nanobots or nanomachines, or thinking about building machines at the nanoscale. By the way, I have links to these presentations, and everyone will get a copy of this because we're gonna fly through it. It'll feel like everybody's drinking from a fire hose today, because my goal is to expose you to as much of this information as possible. But then you can come back and watch it in detail. I'm gonna do this at the end, but everybody would look at this device and go, okay, that's a human-built thing. It's a machine. That is an electron scanning microscope of a bacterial flagella motor. So that's actually what a flagella motor looks like under an electron scanning microscope, which looks awfully like that airplane engine. And these are high resolution computer processing of those EMs and showing actually the individual components of that. And what's fascinating is there's even
15 microns long. Fleegee, the stator.
So there's even science about how these
but Moat B is
flagella get created. So we see a cell wall here. This is the outside of a bacteria, and you could see all of these proteins are basically forming what ultimately is a motor that propels the bacteria forward.
Build up the rod in a proximal to distal fashion. Another set of rings, called L and P rings, are found in gram-negative bacteria such as E. coli. They penetrate the outer membrane, forming a bearing for the rod. As the rod cap is exposed outside the L ring, it dissociates and is replaced by a hook cap that guides the assembly of the hook proteins. After the hook is assembled, the hook cap dissociates, and a series of junction proteins assemble between the hook and future filaments. Finally, yet another cap is built, and filament proteins assemble. Like the rod and hook proteins, they travel through the hollow channel inside the filament to reach the distal end. The cap rotates, which causes the subunits to build in a helical fashion. A complete filament can consist of 20,000 to 30,000 subunits.
Little piece, there's like a Lego brick that's formed from protein that's expressed from DNA. And there's actually even control systems within the bacteria to produce longer or shorter flagella depending on the viscosity of the fluid that the bacteria is flowing through. So there's actually control systems literally to sense the viscosity of the fluid and produce longer or shorter tails. And I won't play all these, it's amazing. Actually, this video here, if you're coming back, shows the math behind and the control systems of how these states how these individual genes interact with each other in order to create, like there's literally math all behind this. So I'm not going to go through this because I'm going to try to keep the math as to a minimum. But there's mathematical equations that show the interaction between the particular genes. And this is a depiction here of the genes and their interactions here. So logic gates essentially in the genes that are controlling, you know, what kind of flagella is built, how long it's built, and all kinds of, when it gets built, and so forth. So we won't go into that, but so how many people know what DNA is in the room? Not everybody's hands. Some people. Okay, we're going to do just a quick thing here on DNA.
Stanley clearly presents, what is DNA and how does it work? DNA, also known as deoxyribonucleic acid, is a molecule. It's a bunch of atoms stuck together. In the case of DNA, these atoms combine to form the shape of a long, spiraling ladder, sort of like this one here. If you ever studied biology or saw the movie Jurassic Park, you probably heard that DNA acts as a blueprint or a recipe for a living thing. But how? How on earth can a mere molecule act as a blueprint for something as complex and wonderful as a tree, a dog, or a dinosaur? To help answer that question, let's first take a quick look at amino acids. Amino acids are tiny little chemicals inside our bodies that are so important, they're often referred to as the building blocks of life. There's about 20 different kinds, each with their own unique shape. The neat thing about them is they can be attached to each other kind of like Legos to produce an endless variety of larger particles known as proteins. Amino acids make up proteins. Proteins, along with other chemicals, combine to form living cells. Cells make up tissues. Tissues make up organs. And organs, when they're all put together and functioning, of course, combine to form living creatures like you and me. These proteins that make up our bodies, and keep in mind, there's millions of different kinds of proteins, they each have to be formed in the perfect shape in order to function. If they're the wrong shape, they usually won't work. That's where DNA comes in. DNA does a lot of interesting things, some of which we don't fully understand, but one of its main and most well understood functions is to tell amino acids how to line up and form themselves into the perfect protein shapes. In theory, if the right proteins are built at the right time and in the right place, everything else from cells to organs to entire creatures will come out just fine. This here is a simplified model of DNA. It shows us that the steps of the ladder are made up of 4 different kinds of chemicals shown here by different colors and letters. If you look at just one half of the molecule, you can read its chemical sequence or genetic code from top to bottom, sort of like a book. A single strand of DNA is extremely long, millions of letters long. It spends most of its life coiled up like a noodle, living inside the nucleus or the centerpiece of a cell. Amino acids, however, live outside the nucleus in what's called the cytoplasm. To help DNA interact with the cytoplasm and convert those amino acids into proteins, special chemicals inside the nucleus make partial copies of the DNA code. These partial copies, called RNA, look a lot like DNA, but they're shorter, of course, and they're missing one of their sides. Their small shape and size allows them to fit through tiny pores in the nucleus, out to the cytoplasm, and into the mouth of another particle called a ribosome. Ribosomes are protein-building machines. They read the RNA code 3 letters at a time, suck amino acids out of their surroundings, and stick them together in a chain according to the RNA code. As the chain grows, it bends, it folds, and it sticks to itself to form a perfectly shaped protein. Every 3 letters of the RNA code tell the ribosome which of the 20 different kinds of amino acids should be added next. For example, CAA tells the ribosome to grab a glutamine. AGU tells it to grab a serine, and so on. Once a protein is built, it can then go on to do a number of different things, one of which could be to help form a brand new cell. So to answer the original question, what is DNA? DNA is a molecular blueprint for a living thing. How does it work? DNA creates RNA, RNA creates protein, proteins go on to form life. This entire process, as complicated, as sophisticated, as magical as it might seem, is entirely based in chemistry. It can be studied, it can be understood.
RNA is transcribed in the nucleus, it goes out, and then it goes out to machinery inside the cell and then produces those little Lego bricks that formed essentially that bacterial flagella motor, right? And the shape of the motor, the shape of those proteins.
Stanley clearly presents, what is DNA and how does it...
So the shape of the proteins form essentially the structure. If anyone see me talk, I always have an a cappella science thing here, and I'm going to play just a little bit of this just to make the point here.
Stated Clearly presents What is DNA and how does it
A dot isn't the best way to try to sum up how electrons come and go. They are the states of a matter field that follows an equation that Dirac wrote. The Schrodinger part of the whole equation will just lead in sub C when it expands. Now get that Coulomb and add it in with the proton and watch them start to dance. It's hydrogen, it's like, oh proton, I feel your tug. Central potential, dip down, pulling on me. But I'm not falling in deep. No, that would break uncertainty. Say, oh, electrons move too much. Slow down your pace and put that orbit on me. Come on now, follow my lead. Come on now, follow my lead. Orbitals take the shape they do, the stable states of the quantum rules. And when a one approaches two, they combine and they're bonding. Cause hydrogen as a rule is found in nature as H2. Energy configuring a molecule, diatomically bonding. Low, high, low, high, low, high, low, high. Diatomically bonding. Low, high, low, high, low, high, low, high. Diatomically bonding. Low, high, low, high, low, high, low, high. Diatomically bonding. Energy configuring a molecule. When orbitals take the shape they do. One half spin will give a lepton a twin. One up, one down in the ground state. With S&P and quadruple degeneracy, the second shell can be filled up with 8. The higher angular hold it true. When orbitals take the shape they do. Come bond with me, baby, come bond. 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 Polymers take the shape they do. Combining base level residues like RNAs, ACGU. With their hydrogen bonding, peptides make a chain and group and get up lead sheets and corkscrews. With the secondary links they fold and move. They're all over your body. Come bond with me, baby, come bond. Come bond with me. They're all over your body. Come bond with me, baby, come bond. Come bond with me. They're all over your body. Come bond with me, baby, come bond. Come bond with me. They're all over your body. You're a chemical machine, it's best to do. The molecules take the shape of you.
By the way, all of that was done without the aid of any musical instruments. That's all his mouth. So I'm impressed by him. But what I want to show is essentially this is a molecular dynamics simulation showing a complex biomolecule and a small drug finding where that small drug molecule will bind. And this whole sequence here that's like, you can see the jiggling of the water and the molecules. This is just thermodynamic jiggling happening. This whole sequence, which is 26 seconds, is only a few nanoseconds in real time. So it's slowed down. Like it's actually happening incredibly quickly. That jiggling is just slowed down for our mere human eyes to be able to see, but it's actually happening very fast. I have a video here that I think I'll just play just a quick minute of it.
Life at its simplest doesn't have eyes or ears, it can't dodge or think or even. Electrons are negatively charged, almost massless and very wavy particles, so when they electrostatically stick to the nucleus, they mostly do it in these stable wavy forms that we call orbitals. Now sometimes it actually turns out that it's more favourable for two nuclei to share some electrons. That is, there is more favourable interactions between the two nuclei and their combined electrons than there is repulsion between the electrons of the two atoms and the repulsion of both of the nuclei. And when this happens, the configuration of two or more atoms stuck together in a stable form we call a molecule. Now these configurations, for simplicity, are typically represented as a line of some sort. Or another way in which we have a rough representation of the envelope of the electron density. Now, all chemistry is basically the nature of which atoms stick together, and given the different amount of positive charge the nuclei can have, and the different number of electrons that they first and third most common element in the electrons are rather unevenly distributed, leading to relatively positive and relatively negative portions of the molecule, and this leads to one of the most important properties of water with respect to life. I mean, let me just highlight this with an example. You see, liquids typically boil according to their molecular weight. So, butane, for instance, is a fairly reasonable gas, while octane, ironically in America called gas, has about twice the molecular weight, and is a fairly reasonable liquid at room temperature. So, acid is like a load of magnetic balls, with a load of thermal motions, which I really can't represent here. But anyway, these magnetic balls stick together very well. Whereas methane would be like a load of non-magnetic balls, which tend to fly apart very easily. So when I get something else that's really quite polar, say for instance like alcohol, and inject it into water, but that only gets you an inside and an outside to the cell. The business end of things is the interplay between mostly two types of molecule, essentially DNA and proteins. Now, DNA basically holds the sequence to make the proteins, including the proteins that actually facilitate the replication and repair of DNA. And it's the proteins that do most of the stuff that regulate what happens in life.
Those forces in order to be able to predict what the outcome is going to be. That's you know, and that can be in one single cell, right? A lot of dynamic interaction, which is one of the reasons that we don't have computers, you know, being able to forecast things. But then now if we move up in the scale here, I want to show, this is an example of DNA here.
Consists of 8 proteins.
Life at
Consists of 8 proteins. The center of each of these structures is composed of a protein called histone. Each center we know that acetylation of specific parts of the histone tails consists of 8 proteins, 2 copies each of 4 types of histones. Researchers have been working hard to understand their roles. This structurally
Show something that's interesting here. This is, every female has 2 copies of the X chromosome in their body. One inherited from their mother, one inherited from their father. Men only have a Y and an X. But in every cell, if you have 2 copies of the gene opened up and expressing, then there's a problem there. So you have to inactivate one of them. And so we'll just play this really quickly here. This is essentially that big strand of DNA. And I'm going to go here. This is inside of every cell in the female body. One is shut down and one is opened up. And so it's accessible to be copied. So information can be read off of it. What's fascinating is, I'll go here, I'm going to go here. There's a whole process for this, which we won't go into now. But when a cell divides, literally, one's from the father, one's from the mother. Essentially, every cell has like a 50-50 chance of whether or not the mom's or the dad's genes get shut off. And then you can actually see, if you actually had X-ray vision and you could see every single cell, they'd be different colors. And so that's actually essentially what in a female, they're effectively got some from their mother and some from their father in every single tissue throughout the entire body. And this is actually, was kind of discovered because they were looking at cats, and it turns out the color of the fur depends on whether the mother or the father's cells got turned on or turned off. So which genes are turned on and which genes are turned off essentially, create what that cell does. And in this case, I mean, color was obvious, but there's lots of other things that happen too. Okay, so that's what we call epigenetics. One of the things I would love, has everybody seen DNA copy? Anybody? Okay, no hands are going up. We'll do this real quick.
Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time.
So DNA
The
is
end
kind of like a
result is
zipper.
two
It
new.
has a certain direction that it goes in, and that's why it has to be copied in reverse on the other side. And it's amazing that these enzymes have evolved to copy and also like do it in a reverse way, right? These are all little robots, machines doing nano machines copying that DNA. And there's some really good videos here that go into much more detail of this process that's fascinating, but I don't want to go into it here today. Another thing you hear is that DNA gets broken. Like if you get radiation coming in like from the sun and things of that nature, DNA can get broken. And then there are processes for repairing that DNA. Oof. By the way, these are all done by Drew Berry. And now it's comparing to make sure that they're all accurate after the fact, to make sure that the two different strands are actually the same. that just blows my mind. It's so fascinating how many tools are involved in keeping that DNA accurate. And all those are enzymes. By the way, everything that you saw that was doing the work there is also expressed in DNA. So in other words, everything you saw that was doing the work makes an enzyme, and that enzyme is created from another template of DNA. So someplace else on the DNA, there's something to call out and build those structures and keep making copies of the structures that can do the repair. This is fascinating here. I'm going to show just a little bit of this. This is an example of another enzyme, another structural protein that allows a virus to enter the cell. So it's called a trimer, and essentially these are just loops here of amino acids that are, again, expressed from RNA and the RNA from the DNA. And I'm going to fast forward here. I'll show you what it looks like. There's two of these that are expressed, and this is the cell membrane, and this is the virus here. It's a very big structure relative to the size of these these entry proteins. And essentially, these are like literally springs that wind up, and when these springs contract, it brings the cell walls together, which causes the viral and the cell wall to fuse. And what's really cool is you'll hear these interferons that they do, these drugs that they give people with AIDS. All they are is a small molecule that winds up and sticks into the spot where the spring goes, and now that spring can't do its job. So when you give these small molecules out, all it's doing is hopefully it's going to the right place, and it's binding into a place that does something good, at least, or stops the virus from working. Often it goes to other places too, and that's the negative side effects of it. But that's what those little small molecules do, is they go in and they bind in. There's another one here, for example, that binds right there, and again, it stops that spring from coiling up and prevents entry into the cell. Gives you a fighting chance. So basically, it's nanomachines, right? If you're into robots, these are all basically robotic functions just at the nano scale. There's actually some cool tools for going out there and exploring these molecules. We have them at Electric Works at our facility, where you can actually use these virtual reality systems and go in and explore and play with the cells and move around and see how they function. And literally just put on the goggles and use the hand motions, and you can literally move around. We actually have these Happily devices that actually let you feel the forces too. So they don't yet. We're actually going to have some students work on actually making these so you can actually try to pull them apart and feel the molecular forces that are going on there. So I made, like, we have all these parts that are working. I'll play another little bit of
a sample scene.
To plan a nanobot, map out the molecules to span the nanobot, add on the motors used to man the nanobot, signals to talk with a commander, software and logic scramble, programmed in nanobot. Beginning, you better control the movement.
The thermal stormer,
Temperature systems are always moving.
your system's moving.
Pieces and links you pick will limit.
The states it can
As it'll randomly shuffle through them. Now force it with the right view. Ordered and energetic, a bias in the setup. Adding entropy through a cycle that'll let it put word to load. Oh, nanobot. So plan your nanobot.
digits, damn. Next, let's see you handle informatics, fam. Nano informatics, the structure of a cache or RAM. Hey, switches are the way to data saving plan. Flip it on, flipping chemically, photochemically, or electrically. Add a flippably, then make a little more deep the decisional tree data. If you need a calculator, how be? Logic gates like A not B. Inputs a cation, that's key. And you get a photon as a reading.
Plan your nanobot to be important info scanner nanobot. Reading and writing coded data that allow freedom to do as you program it. A Turing automatic full gamut nanobot.
Oh, nanobot, doesn't that look like
Oh, there's references for everything that's said
doesn't
there.
that look like Oh,
I think I spent
nanobot,
almost a month
doesn't that look
researching everything. Annotations, literally.
Break me down, I am a nanobot.
Did you notice any here?
The musing model of synthetic nanobots. Working in concert
We'll skip
as a
I give this talk also. Yeah, go ahead. There's a question.
For all I try to be more than it, though sentient and organic, I am a nanobot.
Oh, nanobot,
That all of us are this way. Your question is, are we all basically nanobots? Yeah, I mean, it's informatics all the way down. I have some slides that are going to blow your mind in a few minutes that might address your question. I don't know that I want to create an existential crisis in the room, but yes, there are some questions about what we are, is what our bodies are, that I'll address. Yeah. So good, that's where it tends to lead to bigger questions about. What I love about this is the problem is we need informatics in order to understand ourselves, biology, and understand the fundamental processes of aging and disease. But it can be understood. That's the exciting thing. It's not woo. There are fundamental rules that can be understood, applied, and then you can actually cure disease. We can hopefully extend life and the quality of our life. So that's one of the reasons I'm excited about this. But good question. We have a bunch of computer scientists in the room, no biology people, but I usually educate the biologists in the room about how computers and informatics work. Oh, we do have a chemist. All right. Well, I'll show you you can definitely go back and watch this, but essentially a lot of what the DNA is doing is what computer CPUs do, right? A Turing machine is essentially reading from a tape, just like DNA is a tape. So, but yeah, so there's a lot of parallels there. This is a wonderful simulation here.
Throughout this
I want to play just a few minutes of this.
This is particle life. It's a very simple particle system that creates these incredibly beautiful lifelike structures. It can be explained in a few seconds, and if you know how to code, you could write your own version of it today. It's a perfect example of emergence, the phenomenon when many simple small parts make up a larger structure that suddenly shows complex behavior. This is particle life. It's a very simple particle system that creates these incredibly beautiful lifelike structures. It can be explained in a few seconds, and if you know how to code, you could write your own version of it today. It's a perfect example of emergence, the phenomenon when many simple small parts make up a larger structure that suddenly shows complex behavior. And then red should be repelled by green. I will explain how
just put
all
in
of this
rules.
works
Red gets
in the
repelled or
second half
attracted
of
by
the
green.
video. But for now, I will just leave you with a few beautiful impressions of particle life. For our new structure, each color should be attracted to itself and to the next color. So red watch what happens.
And then red should be repelled by green.
And they start following each other.
So I think you get how it works. Let's try to create a new structure. For our new structure, each color should be attracted to itself and to the next color. So red to orange, orange to yellow, and so on. To try this out, I placed all of the colors next to each other. Let's just watch what happens. You see immediately all the colors clump together, and they start following each other.
ParticleLife.com, you can put your own rules in and get your own organisms, or what appear to be organisms. And so there are these just self-organizing structures, and like we all know, like these are just highly complex. These are swarms of drones, right, doing
All
shows.
of that snake-like movement just emerged
following
from these
code.
simple
And
rules.
but what's cool is a lot of these drone shows.
Take a look at that
if
in
a
slow
drone falls
motion.
out of the sky, they put swarm logic in so another drone can take its place, right?
You can
So
download it from this
you basically
website.
build these small rules about how that show should, if somebody falls out, who should take its place. And yeah, this is an interesting one. These drone swarms.
These are boxes full of drones.
News, New Jersey was flipping out with drones in the sky. This is the kind of stuff that was probably causing those issues, right? So, and again, by the way, I'm not going to go too much into the math.
A blue sky Tennessee day.
Who knows what a PID
But
controller is?
there's a storm coming.
One? Because these are robotics.
Look a little
the first
closer,
robotics.
and it's a city under siege.
Yeah, exactly. Oh, it's electrical engineering, right?
Piercing
So
the air
a PID
around the
control
Fort Campbell
is
base,
a
a mob of
proportional,
drones on the hunt.
integrative and derivative approach.
There's nobody around
for
here. The
control
city's
theory.
been constructed by a US military
and
experimental unit to see how swarm.
This is what's called a PID controller, where it's sensing. You're using a part of the air. In other words, your goal is to keep that ball in the middle, and you use, PID is proportional, I is integrative, and D is derivative. So you take the error and you take a proportion of the error and you move. You basically move the arm
with the
a
tests.
proportion of the error, meaning there's amount of
In
error.
the
You want
code
that
on
thing
this
in the
part,
middle.
you will see the KP.
And that's obviously, now that's like flopping back and forth. So if you always you overcorrect, and so you can overcorrect and you never get to the middle. And essentially you add integrative error and you'll see that it does better. So these are set to 0. This is derivative. This is using the derivative of the error. It takes too long to get there. But when you use all of those together, you can get a control system where when you're measuring the error here, it immediately responds and gets you to a stable state. And that's a PID controller, right? And this is what's used in robotics and any kind of control theory. There's other versions of PID. PID's kind of the fundamental, the first thing you learn, but there's other ways to do it. In fact, your thermostat uses a kind of PID controller for commercial, and there's actually PID controls within the bacteria, by the way. So oddly enough, like we're thinking we've invented PID control, and it turns out it's in our DNA. Like our DNA has, and not just our DNA, all of life's DNA, on how to actually move bacteria to get where they want to be near food, to chase food, and how to control the bacterial flagella motor. It's using a PID controller in order to hunt food. And you can actually go into, he's actually got a beautiful story here showing how often you tumble is based on the chemical sensing of certain proteins and signaling pathways within the cell. It looks complicated, believe it or not. Like if you watch the whole video, it does a very good introduction of PID control within bacterial movement. And he does a great job of building that up. So I highly recommend watching that and some of the other ones if you're not familiar with PID control. about fundamentals, I want to bring up another level, which is evolution, right? So that's control within the cell, but there's also evolution going on as well. And what I'm gonna mute it here. So this is actually a really cool mega plate, a big, huge petri dish. Essentially, what they did is they took bacteria and they put different levels of, antibiotic. So no antibiotic, some antibiotic, 10 times the antibiotic, up to 1,000 times the antibiotic here. And then essentially what they do is they put bacteria into the dish, and the bacteria love it. There's no antibiotic, and you could see as soon as the bacteria get to the antibiotic, they slow down their growth. They can't, the antibiotic is stopping them, but all of a sudden a mutant forms that's well suited to that small amount of antibiotic, and now those cell lines of that bacteria divide. And then you could see, boy, it took them a little while to get into the antibiotic, but then, boy, getting into 10 times the antibiotic, it didn't take very much. But then you can see getting into 100 times the antibiotic, and you're watching essentially bacteria develop antibiotic resistance over a number of days in a petri dish. And what's really cool is they also can now go back and map each individual mutation that occurred in the bacteria, and you could go use. We have one. We're building one at Electric Works right now. Literally just a CNC machine that we've modified to go take out and sample cells off of a plate and then go put it into a sequencer. So we want to be able to do this experiment ourselves at Electric Works. So what's amazing to me is that the bacteria are evolving. In other words, the DNA. By the way, actually, sometimes DNA doesn't change. It just turns on or turns off certain expressions. But there are interesting ways to actually cause DNA to change, intentionally change, in order to express slight variances. And those variances turn out to actually, if they confer survivability, then they get expressed in the next generation. Okay. And by the way, this is one of the things that I love to study. These are embryos. This is a fly embryo. And what's amazing to me is right around, all the cells divide for the first 2 hours, and they're all copies of themselves. But then they get to a point where they're like, okay, we have enough of ourselves. We have enough cells to actually go to the next stage. And then boom, just like a marching band, they all start to play a different song, and they all go to some other place. The cells each individually differentiate and express different kinds of DNA in each different cell. And that's how you get different tissue types. And Eric here built some software to actually track and map each cell and its movement. So you can see basically every cell in its position, it moves, and you can actually see it colorized as to where they go, which is really cool. Does everybody know who the Mandelbrot? I have a Mandelbrot tie. How many people know the Mandelbrot set? All right, good. We won't spend time on the Mandelbrot. But what's really cool is a very simple equation can produce so much variation, just depending on the location that you're at and the sequence that you're at. And one of the areas that I like, what we're studying in our lab down at Electric Works, is C. elegans. So C. elegans is a worm that has always 980 cells. The adult has 980 cells, and this is what the worm looks like. And there's actually, what's really cool is because it's a simple model, it's studied a lot for cancer, for neuron formation. This is a computer software that actually simulates every single cell within the worm. So that's what this open worm project is. And you can actually go in open worm and literally go around and browse around and actually see every single cell, every single structure, and how it's mapped to the next one. It's pretty cool. What I'm interested in is understanding how they form. So this is actually that worm from 4 cells, and then into 8 cells, and then into 16 cells, and then 32 cells. And we have the equipment at Electric Works to be able to look at this under the microscope and also do individual gene sequencing. So does everybody know who Michael Levin? Anybody know Michael Levin? No, okay. Michael Levin is a researcher at Tufts University, and this is a fascinating experiment. So this is a flatworm here, and I'm actually just going to play this just so you can hear it.
They're known as a planarian. They're flatworms. They're actually quite a complex creature. They have a true brain, lots of different organs, and so on. And the amazing thing about these planaria is that they are highly, highly regenerative. So if you cut it into different pieces, in fact, over 200 pieces,
what's going
every
on
piece
here.
will rebuild. This is a creature known as a planarian. They're flatworms. They're actually quite a complex creature. They have a true brain, lots of different organs, and so on. And the amazing thing about these planaria is that they are highly, highly regenerative. So if you cut it into pieces, in fact, over 200 pieces, every piece will rebuild exactly what's needed to make a perfect little worm. So think about that. This is a system where every single piece knows exactly what a correct planarian looks like and builds the right organs in the right places, and then stops. And that's one of the most amazing things about regeneration. So what we discovered is that if you cut it into 3 pieces and amputate the head and the tail, and you just take this middle fragment, which is what you see here, amazingly, there is an electrical gradient, head to tail, that's generated that tells the piece where the heads and the tails go, and in fact, how many heads or tails you're supposed to have. So what we learned to do is to manipulate this electrical gradient. And the important thing is that we don't apply electricity. What we do instead was we turn on and off the little transistors. They're actually ion channel proteins that every cell natively uses to set up this electrical state. And so now we have ways to turn them on and off. And when you do this, one of the things you can do is you can shift that circuit to a state that says, no, build 2 heads, or in fact, build no heads. And what you're seeing here are real worms that have either 2 or no heads that result from this, because that electrical map is what the cells are using to decide what to do. And so what you're seeing here are live 2-headed worms. And having generated these, we did a completely crazy experiment. You take one of these 2-headed worms and you chop off both heads and you leave just the normal middle fragment. Now keep in mind, these animals have not been genomically edited. There's absolutely nothing different about their genomes. Their genomic sequence is completely wild type. So you amputate the heads, you've got a nice normal fragment. And then you ask in plain water, what is it going to do? And of course, the standard paradigm would say, well, if you've gotten rid of this ectopic extra tissue, the genome is not edited, so it should make a perfectly normal worm. And the amazing thing is that that is not what happens. These worms, when cut again and again in the future in plain water, they continue to regenerate as 2-headed. Think about this. The pattern memory to which these animals will regenerate after damage has been permanently rewritten. And in fact, we can now write it back and send them back to being 1-headed without any genomic editing. So this right here is telling you that the information structure that tells these worms how many heads they're supposed to have is not directly in the genome. It is in this additional bioelectric layer. Probably many other things are as well. And we now have the ability to rewrite it. And that, of course, is the key definition of memory. It has to be stable, long-term stable.
And in fact,
it has to be
in
rewritable.
this next talk, which I'm not going to play, he talks about like taking cells that are cancer cells, like intentionally causing the genes to be cancerous, and then using electrical mechanisms to drive them to be non-cancerous. So like basically turning off the cancer part. This video, like your question earlier about what are we and what is intelligence, these videos here, if you watch them, they'll give you a better perspective on that. But one of the things I want to show.
It's not overtrained on its evolutionary prior. What it does is it
just a cell that has multiple copies of the DNA in it. You literally just jam more DNA in it, and that
In
means
many
that
ways, biology
because there's
does
more
not
DNA, for
overtrain on
whatever
its
reason, the cell
evolutionary prior.
becomes
What
bigger.
it does is it plays the hand it's dealt. And what I mean by that is, here's one example. This has been known since the 40s. This is a cross section through the kidney tubule of a newt, and typically newts have like 8 or 10 cells that work together to make this tubule. But if you force the early embryo to be polyploid, and you can get 2N, 4N, 6N, and so on, what happens is the cells get bigger.
Every cell is going, it's got a job to do. What's it supposed to do? Well, in this case, these cells are supposed to form a tube inside the what was he saying?
That's pretty
talking?
cool.
Oh, inside of the kidney of a newt, right?
Cell
The kidney is where you're going to have urine
until you
come
make
out.
really
So pretend that there'd be
gigantic
urine here.
cells,
These
where
cells'
now
job is
instead
to
of
go
using
do that.
cell-to-cell communication,
And
a different
if there's,
molecular mechanism
the
is
cells
called
are
up
bigger, they just
to
form the
reach
same
the
size,
same
but they just
anatomical outcome.
like,
This is a kind of
do it with
top-down
less. And of
causation,
course, if there's
where here
so
now
big
cells
that only
use the
one cell
cytoskeleton
gets
to
to do
bend
it, then
around
only one
themselves,
cell
leaving
does
an
its
open
job.
hole in the middle.
And we
these are
don't
5N newts.
understand this.
So
This is an open area of research as to how do the cells know what to do? How do they know what role they're supposed to play? And there's so much to actually understand and learn about this.
In many ways, biology
Actually has a whole thing about consciousness and where does it emerge and emergence and everything else. That's a fascinating to hear him speculate. So I put that video in there. Yeah, question.
So this kind of implies that for occasionally in biology, if you give enough input into something, like it dramatically changes that.
If you're a cell, in many ways, biology
one
does
thing
not
that
overtrain
people
on its
often
evolutionary
think
prior.
Yeah,
when,
so
when they
his
hear me
question
talk about
was, you can dramatically change it by giving input. Let me show you guys. It's fascinating. There's these things called galls. Here we go. Is that it? So this is used in life all the time to reprogram tissue in plants. Here, actually, I'll do this. These are called plant galls. Let's do this. We'll do images.
In many ways,
That's a
biology
good example of one. So that's what's called a plant gall. You'll see these beautiful structures. And what happens is a bee goes in and lays its eggs into the plant, and the bee, the larvae actually signal the plant. All the growth there doesn't come from, it comes from the plant. The plant's now being told to change its structure and what it would produce, and it defends the growing embryo of the bee, and all those cells change their function, and the cells are actually plant cells. Those are not embryo cells, those are plant cells forming a protective structure around that little embryo. So yes, like every cell could be reprogrammed to do something else if you knew how to talk to it. So if you're a cell whisperer, you can tell the cells to do something else. So, by the way, people think this is kind of fun. I love this one here. Cells do their programming. So this is a neutrophil chasing bacteria. This is a real-time thing here saying, okay, everybody understands their immune system or chasing, just like bacteria have this flagella. These use microtubules to stretch and move, but they're following that bacteria till they're able to eat it. And by the way, if cancer evolves, it can actually metastasize, and that's how cancer can go and get the function to be able to move around and actually go around your body. And that's one of the functions of metastasizing, is to activate the genes that give you the motility to do that. What's amazing to me is that is also, these are neurons moving around. So if everyone thinks their brain's kind of a static thing, no, especially during the formation of your brain, these are neurons using the same tricks to move around and find partners to communicate with. And so they have that, there's a motility mode that a cell can kind of pick up and start to go, okay, I'm not going to sit here anymore and do my job. I'm going to go up and change my internal structure so they can stretch and move and reach out and do things. And you can see how it's actually like What's funny is neurons want buddies. They always, neurons want to wire together and fire together. Let me just play a little bit of this. I got like 4 minutes I want to do.
Hi. Huxley. Be back. Oh, Carol. Goose. Stephen G. Davidson and Peter. Go. See, one cell divide and decide on 1,000 fates. Did you ever figure how they know? Be back. We are built of modules combined in a planned out way. Each new piece must be told where to go. Oh, now there's a science helping us to understand how our cells encode this architectural plan, signaling each other with genetic tools. Oh yeah, wow. Phenotype the interface for mouse and man, genotype the files and the subprograms. What then are the switches, circuit boards, and boot code? Devo, looking at the logic in the ways that we grow. Every gene directed by a signal key code. Proteins that can activate, enhance, or veto. Devo, the signals are controlled by other genes that signal. Calculating in a network labyrinth, though, where the heart and liver and the hands and feet go. Signal mapping tells each region what it ought to be, yo. With circuits so deeply built upon, they're older than the Paleo. The Paleo's a wicked baby. In a crucial pathway, changes tend to get torpedoed. Where they go, calamity goes. As the cycloning shape knows. See down, they cascade like a domino. Like you and I, Drosophila. The path that makes us optical was laid a long, long time ago. Back before we blew up the Cambrian like a bomb. Now my eye protein can make you see out of your bomb. And hedgehog and its relatives like Indian and Sonic. Set up in a gradient on segments embryonic. Split for brains and asymmetric parts depend upon it. Flipping on genetic switches and logic. From Devo to Evo, adult and embryo. Mostly don't evolve in the genes of the genome. Save for the mutation in their regulation. Keep the building blocks and swap their activation. From Devo to Evo, parts of alter egos. Homologs evolve from repeats in the schema. Switch a couple bases in the proper places. You'll be watching flies grow legs out of their faces. Oh yeah, Evo, Devo. Stick around for modern synthesis, the sequel. Only by combining can a new theory grow. Evolution and development, amigos. Evo, Devo. The signals trigger patterns of complexity. So switching up the switches of a signaling node gives a modular and simple way to evolve. Switches. Look at our spinal sigma generate an hydro. Built on a molecular cloud. One single, one vertebra. One vertebra, baby. Speeding up its radius, snakes developmental cheat code. That are where a lizard's feet grow. They turn off distal aminos. Evo, Devo. This is how we go from single cells to people. Every generation and in life primeval. Life in variations and the sand beautiful. From Devo to Evo, larva to mosquito. Patterns are resolved as the signals proceed. Never gene with the glow tag. Kill it with the morpholino. Shortly go morpholino. From Devo to Evo, voyage of the beagle. Body plans evolve when proteins steer the gene. Better life's beauty grows. Aesthetic in vivo. Evo, Devo. Acapella science.
I love this stuff. So what's really cool here is this is called the homeobox. They discovered a sequence of genes that the order in which the genes appear actually lays out your body plan. So if you literally swap things in that particular region of the genome, it will change the body plan. So this is the homeobox right here, and there's proteins in a very specific order. And it's very well conserved. In fact, the explosion of the paleo, like the Paleolithic era where a lot of mutation happened, it's thought that there was a lot of variation happening in the homeobox, that for whatever reason that mutated very quickly, which created lots of different life forms and lots of different body plans. It's a theory. But what's really interesting is that if you just take proteins during development and you change them ever so slightly, you can get different body structures, or when he said the eye out of his eye protein here, yeah, like this is the,
Long time ago, back before we blew up the Cambrian, the K-bomb bomb. Now my eye protein can make you see out
So
of your
literally, there's a protein that if you put it into the embryo at a certain time, that's where the instructions were, and now an eye will form, and that eye can actually connect up to the. Literally, the body will vascularize it, like give it blood, and it'll actually connect it up through neurons to the spinal cord, even. But you may or may not be able to see out of it, like in terms of what that organism can see. So literally, just finding these little sequences in the genes and understanding the body plan. Yep, there's a question. Yeah. So his question is like talked about it being different than mutation. Like for example, we know that there are instructions in our bodies that can build gills. They're just turned off. So like you can almost trace the whole evolutionary lineage of an organism because you'll see parts of the DNA that are still in your DNA that just never get expressed. So there's a bunch of DNA that you have that doesn't even get expressed. But it might get expressed. It might get turned on under certain circumstances. Does that help answer your question? So one of the ways, there's a really good book called The Selfish Gene by Richard Dawkins that talks about this in particular, that your genes are not survival of the fittest of the organism, it's the survival of the fittest of the herd. So another analogy I would give is that everybody here has a gene that controls whether we're early morning people or late people. Like every person has in themselves genes that say, I'm gonna be an early riser or I'm gonna be a night owl. But it turns out for the herd, some people will express and become early morning risers because you want some people within a herd, if there's, you're trying to protect the herd, you want some people to be awake during the morning and you want some people to be awake at night. And there are odds in the genes that actually say how many people will express one gene or another. And in his case where the butterflies had the spots, the idea is that all butterflies could form spots. But what you do is you turn on some butterflies that form spots and some butterflies that don't form spots. But they have the genes to be able to do it, but you're swapping the activations. You have certain activations that turn them off and on. Did you have a follow-up?
This is going to sound like a little bit off the tangent, but so there's this cartoon series where the basic idea is this one individual, he creates a society, then every 1,000 years he creates some kind of chaos
that shakes up the population. Yeah.
For some of them for a lot of them to die, for the majority of them to evolve.
Right.
Like they give an analogy of Atlantis. He literally made the pact with some deity to sink the city because he knew that there was a chance because of all the chaos he's caused in the past that some of them would evolve and be able to live on.
Yep. What's your question? Is that just? Yeah, so by the way, this book, The Selfish Gene, that I talk about is really good. In 1972 or 73 it was written. There are these strategies called, you have to think of our bodies, like for example, you have bacteria that are in you right today that you wouldn't survive without the bacteria. And we are all in what's called a prisoner's dilemma. If you Google prisoner's dilemma, does everybody know what a prisoner's dilemma is? Yeah, a prisoner's dilemma is we're all in a prisoner's dilemma with bacteria, meaning we're cooperating. They're actually giving us vitamins and we're giving them food. But as soon as you actually die and you're not giving them food anymore, they defect and eat you. That's partly what decay is, right? They switch to another mode and they're like, okay, we're not going to cooperate. We're going to, so, that happens in societies too, where we've evolved the ability to be collaborative with each other. And so a society can't, there have to be in any stable society, there have to be a number of thieves, a number of liars to keep everybody else honest. Like there's a herd thing that has to happen, because if you have just everybody who's just, doesn't know how to lie, there's actually a funny, The Invention of Lying. There's a whole movie about this, a comedy about the invention of lying. If we all all told the truth, then as soon as somebody evolves lying, they'll take over the entire population, and the population won't survive because the liar will be able to take everything from everybody, right? So you need sort of these pressures that happen within a herd or within a dynamic to actually challenge the rest of the herd, right? And that's what what your story was a population where in order to keep the population healthy, you have to sort of challenge the population. These are very similar ideas, if that makes sense. So I'll definitely be able to talk more about that with you if you're interested, but we could do that after. By the way, the cool thing is that we can experiment with the DNA. I want to show just a little bit of this. Here's an example. I'm going to mute this. So this is actually a cell nucleus that's stained in red, and we have the actual equipment at Electric to do this experiment. And what he's doing is he's tracking a specific RNA inside the cell nucleus. We learned earlier that the RNA is red in the nucleus, and it's just flopping around thermodynamically looking for what's called a nuclear pore, a hole to exit out of. And what he's done is he's mutated that protein to have what's called GFP, green fluorescent protein, that's embedded in it so that we can track its movement. And so it glows green when you hit it with a green laser, and you can actually find out where it's at. And we have a confocal microscope with a N-spark detector. It's the fastest one around in Electric Works that we can recapitulate this experiment here. But what then he does is then he then tracks where the RNA gets out that nuclear pore and it's moving around. These are the tendrils, if you will, of a neuron. These are the axons of the neuron as it's moved. I showed you earlier it was moving around and doing things. This is showing these RNAs moving around, and he's able to find out when they stick someplace, they form a new branch. So he's literally tracking that RNA, where it's going, and you can see it's kind of just moving around looking for a place to go. And we actually have, that was, I can't remember when that was probably 10 years ago, but he was able to show that this particular RNA sequence was fundamental in forming a synapse with another neuron, and the idea was this particular RNA or this particular gene that expressed this RNA is the basis of memory, of forming memories in your brain. And so it's just absolutely cool that we can now track each individual RNA moving around in the body. I want to show just a little bit of this. This is synthetic biology here, and then we'll just do Q&A.
And if either is on, where they're both on, then the output is off. So believe it or not, that simple function is all that's required to build anything that you see in the computer. So now, that's sometimes hard to wrap your head around. So I like to use the example of the Apollo 11 missions to the moon. These were very basic computers at the time, and believe it or not, they were entirely based on interconnected NOR gates. So it took 5,600 NOR gates in order to create the guidance systems that took those astronauts to the moon. And so if we had 5,600 NOR gates that we could put into a bacterium, we could reproduce the Apollo guidance systems within a cell. So the trick then becomes, how do we create genes and then create it synthetic?
These are the names of genes here and how they interact with each other. So a gene, when it gets expressed, can go over and silence another gene. So genes actually interact in this complex way of logic. Some genes actually turn on other genes, and some genes turn off other genes. And he basically built a map of which genes do the interaction here. And then the cool thing is he went in and mapped all of those interactions here and how they interact. So they're chemical, they're probabilistic interactions. And then he built a product right here.
This is software. This is a movie show. This is software. This is a movie showing how the software actually works. So you go into it, and you first have to set the sensors that you want. So you may want your circuits to sense a small molecule or oxygen or a metabolite or a communication signal from another cell. And you can either use the ones we have or upload your own. You then write the circuit function that you want using this Verilog language. And so here we're just showing a very simple logic function that's being written.
The programming language you build processors with. So if you wanted, that's what they do for graphics cards and for CPUs. If you want to build a CPU, you use Verilog to do it. So literally he took the exact same language that you use to build CPUs, and if I back it up here and I show you right here, you're putting in these particular things, and then there's the promoter sequence. That's the sequence you need in order to promote another expression. So
signal from its Verilog language. And so here we're just showing a very simple logic function
Verilog
that's
is what
being,
you'd use to
written.
build a CPU
And then you go in,
logic.
and there are a couple things that you can specify. So you choose the organism that you're compiling this, and then once it's run out, and for all the different states of the circuit, which sets of repressors are being produced. It's making sure that those response functions for all of those gates cross each other nicely, and thereby making a nice overall circuit function. And then finally, it actually predicts the experiments. So this is showing the distributions that experimentalists would get if they ran a method called flow cytometry that allows you to look at tens of thousands of cells. And then finally, this is the sequence that comes out. And so this is in NCBI format. And this is the DNA sequence that then encodes the program that was written a few seconds ago.
Literally, and you can go
And so
synthesize that sequence and then stick it in a bacteria and then turn the bacteria into doing what you just asked it to do with that code. So it literally tells you these things in bacteria. They have simpler DNA, not the big, long DNAs. They have a thing called plasmids, little circles that gets bundled in, and you just stick that into the DNA, and now all of a sudden that bacteria does your bidding. And there's all kinds of things you could do right now, like build cells that go out and seek. And one of the things I'm interested in is, okay, go observe what's going on inside the cell, translate that back into DNA, put it into a vesicle later, and then go read back what that cell observed as DNA, and then be able to essentially build biosensors that are inside your blood, reading that out as DNA. So I do want to tell everybody that one of the things we're going to do next year is iGEM, the International Genetic Engineering Machines. This is a big conference where it was started at MIT. Let me just do this again. So you can see it's essentially like FIRST Robotics. For anybody who's done FIRST Robotics, it's a big competition where you build robots in high schools. This is mostly for colleges. So next year, I'm going to be funding as many teams as I can get started here in Fort Wayne from the colleges. But this actually was started like back in, when was that? I think this was, well, this is the most recent one, yeah. Actually, I think that's a link I can take. We'll come back to it. This is a history of iGEM, and you could see all the teams. This is for 2021, and where they're all over the world, North America, Europe, Asia. There's a huge number of teams in Asia. And Purdue down at West Lafayette in 2020 took gold for building, here I'll play just a little bit of this. Essentially, they built a microfluidics system.
Point-of-care rapid COVID-19 saliva-based diagnostic device.
And essentially, it's a little lab on a chip.
This works like this. Saliva is inserted into
in the presence of COVID. So literally the liquid gets pulled in here and into this section, and it reacts here, and there's all these steps that come through with different reagents that eventually just glow. Oh yeah, they glow red if there's COVID there. But it's all doing, instead of using chemistry, it's using biochemistry to do that. And they took gold back in 2020. This was a China team that did anti-aging with CAR T therapy. So they were using CAR T therapy to actually prevent aging. So it's pretty cool. And this is some details about the sponsorship. So I'm very much interested in getting both the biology and the computer people to work together, because technically the biology is informatics. You know, it's just the study of the information, and I want the biology people. I was up at Trine University studying, touring their facility, and on the first floor they had all the computer stuff, and on the fourth floor they had the biologists, and the computers were playing chess. They were moving pieces on the first floor, and I went up to the fourth floor, and I saw the biologists holding their hands and like pipetting and struggling to pipette because their arms were tired. I'm like, we have to get all these people in the same room. The robot should be running the pipettes, not the people. And I'm a big believer that we need to cheat. I love ethical cheating. I assume everybody knows Mark here, but he's a scientist who likes to cheat. So this is one of the things I show is scamming these games here by building things, right? So I want to cheat biology. I want to cure disease. I want to cure, old aging, and that's what we're doing in biology and in synthetic biology. So if you're interested in joining, there's Drew over there. There's Drew right there, yeah. So he's the person to get there. And I'd love it if Purdue Fort Wayne started a team and did both, did bioinformatics and actually did do iGEM. So now I'd love to just do Q&A here for the next 15 minutes. And are there questions online, Drew? Do you know? Nope. Okay, good. Anybody got questions? Yeah.
For those of us that computer science students that don't necessarily have as much of a scientific background, how do we, once we get lost, if we try to help out with projects like this?
Your question is computer scientists that don't have the background in biology. Yeah, chemistry and biology. The cool thing is that you don't need to know the chemistry and biology necessarily. So here's a fun thing. We'll do this. Oops, let me do this. Yeah, 03 is good. I'm picking my model. I'm trying to figure out which model to do. Write a Python script to download the DNA sequence of a fly and compare it to C. elegans. I would say use AI. Like AI will help you. So there's bioinformatics. The module seems useful for downloading the DNA from the MCBI. That's the national. There's a National Center for Bioinformatics, is NCBI, and it's thinking right now. I should have done the fast one so it would go faster. ChatGPT will do you wonders, and it'll help guide you through it if you explain it. So now it's going to build a Python compare genomes. Did it write the script? Oh, yeah. I scrolled. There we go. So right here, right? So it's just importing NTRES and seq.io, from bio, and then also bio-seq utils. It's fetching the sequences. A lot of times it's fetch nucleotides. So you have to go in and look up. It doesn't know the organism name here. It doesn't pull it in. So there's some things you have to help it with. Just like you're doing your homework, you can come through here and it will help you do that. Like I honestly think the use of AI will accelerate your knowledge. You can even ask it questions. I've learned so much. I've learned so much from just using ChatGPT, to help me through that process. That's one of the best ways to do it. I would say like that, if you're interested in it, go. I've tried to lay out. One of the things I've been here is tried to be a docent that builds the knowledge and the fundamentals. But there's millions of questions you could. Just even going on the fundamentals of this slide, and you don't necessarily need to know the chemistry in order to get there, right? There is just some basic informatics level, just like emergence, right? Like we don't need to understand quantum mechanics in order to have laws of economics, right? There's economic laws which ultimately are based on thermodynamics. I got the chemistry guy up here. I'm going to be in trouble, but. Or another example is thermodynamics is based on quantum mechanics. So there's all these rules on thermodynamics that you could just work at the thermodynamic layer, right? And you don't have to understand the quantum mechanics. Now there's going to be regimes where the thermodynamics doesn't work because you got to understand the quantum mechanics. There's edges to where those rules are applicable, but that happens in biology too. Like you can just understand the higher level stuff, and then later you'll pick it up as you need it. Is there a point there? Yep. Yeah, or take a course. That's a good one. But I will tell you, it's often like you can get sucked into within a course of like deep academia that maybe you don't Like I actually, when I went to Purdue here myself, and I think I had to take thermodynamics twice, and I hated it both times because I got a D in it the first time and I had to retake it and I hated it. I didn't actually like thermodynamics probably till 10 or 15 years after I graduated, and then I took it again and I freaking loved it. Like I was like, oh my God, why didn't I pay much more attention to it? It wasn't, and I only took it because I wanted to understand these binding energies of these molecules, right? And then all of a sudden it became more interesting to me, and then I wanted to apply it. So I'm a big believer you learn what you need as you go, and there's awesome, like the funny thing is if you just type YouTube and whatever you want to learn and then you start sorting it, you can almost find everything on YouTube. But if you want to understand it deeply, then you take a course. Yep, question up there. Oh, great. So you were bored. You were bored the whole time, so. Thank you, thank you. So, great. I love volunteers from the teachers wanting to get involved, so that's great, that's great. Yeah, question?
I was thinking, going back to that Evo-Devo video, I was fascinated by that process of manipulating proteins to grow organs where normally they don't. And I was wondering if we could, if we found a way to control that process. It sounds like that could lead to major breakthroughs in genetic engineering. Do you think that that's something possible in the near future?
Yeah, that's what synthetic biology is. That's actually what, going and manipulating the genetic code, which is fascinating because it turns out a lot of it may already be there. In fact, one of my talks was like I was saying, hey, there's plenty of room at the bottom and we'll make nano machines, and we're thinking, okay, we've got to build these motors. Well, as soon as we start to get to that level and see it, we're like, holy moly, there's already nano machines here. We don't need to build it. We just need to use what's here already. And what we'll find is in the genomes, we'll find that the genes are probably already there. It's just how do we activate them and how do we control them in a very complex symphony? How do we like turn up the music on this one particular thing and turn down the music on this other? So no, absolutely. That's something I'll say, like every single cell in your body, there's this thing called the immortal cell. It's a great book. And there's these cancer researchers who are basically trying to understand cancer. Now, cancer has this amazing ability to express so every time every time DNA copies, there's these things called telomeres. Everybody's probably heard of telomeres, maybe, hopefully. Telomeres are like the little eyelet on your zipper that when you start your zipper, you zip up, right? Well, what's interesting is the DNA polymerase can't copy the eyelet. Every time it does that, the eyelet falls off. You have so many eyelets it could do. And so every single cell has what's called a Hayflick limit. This is Guy Hayflick who discovered that when you're culturing cells, you can only culture them so far and then they won't copy anymore. So most cells are what's called the somatic cell. Or the somatic cells are mortal cells, they die. And there's the immortal cell. Like every time you form a new cell, a little embryo, that's immortal. Like it's going to live forever, and then it will then divide and create children. When you have sexual reproduction, that cell will be immortal. That gene, that pathway of that species will be immortal. But most of our bodies are mortal. One of the things, so does everyone know what biofilm is? So biofilm, they talk about it in hospitals all the time. Biofilm are bacteria. When bacteria divide, they form these complex structures that make it really hard to kill bacteria. And it's thought that that's the first bodies, the first multicellular organisms. And what happens is that when the bacteria is under stress, either by viruses attacking it or by chemicals, it divides and it makes copies of itself, and it does it for it makes copies that are basically going to take damage, and those copies don't replicate. So it's making, I think of it like, I don't know, like a what was that superhero? Marvel Comics, right? Where he copies himself and then the copies fight with everybody, and then all of a sudden the copies go away and he's still alive, right? So Dr. Strange, that's the one I'm thinking of, right? Literally bacteria basically evolved to make copies of themselves that then take damage from the environment in order to protect the immortal bacteria. And it doesn't want to compete with those bacteria for food. It's just making copies of itself. And by the way, it also copies itself, and it's like, okay, I'm going to make a copy of myself, but I'm not going to put all the effort to make a complete copy of myself, because a virus is just going to go into it and then attack it. So I want it to die. If any virus that goes into this little bacteria is destined to die. And that's why it makes copies of itself, so it can take the damage, go away, and then it can skate away scot-free and then go on and live another day. Essentially, our bodies have evolved in that way. Our whole body is the somatic cells that are there to protect the germline. And I joke, I posted once on LinkedIn that I'm the somatic revolution, the somatic proletariat rising up against the gene bourgeoisie, the genetic bourgeoisie, right? The bourgeoisie are the germline that keeps propagating. But my whole body and my brain is there literally from an evolutionary standpoint, from a copying standpoint, is there to protect the gene line. But I have a brain, I have memories, I'm a sentient person that wants to live on, but all my cells are programmed to die. Most of the skin cells die within 14 days and are replaced. Your intestine is every 3 days. Almost all your cells are all programmed to die. And that's by design, right? If you have a virus, a wart, for example, that gets into your skin, that's a virus trying to use your machinery to reproduce itself. And it's best if those cells don't, like if it goes in there, it's captured and then it will die, it'll be sloughed off. If you injure yourself, you cut yourself, most of the time healing is the process of stopping the bleeding. And then normally your body's gonna replace that tissue. Anyway, like your process of healing was those skin cells were gonna go away anyway. They're dividing in a consistent way to get rid of it. So healing is just the process of replicating and replacing and keeping homeostasis, like basically keeping you. But most of your cells have been replaced. Fat cells are like every 11 years. The longest living cells, by the way, in your body are your neurons in your brain, it turns out. But most of the other tissue that's in your hand, like your hand is like an anthill. It's constant, like the ants are constantly being replaced, and your hand is replaced. Most of your bone is remodeled and replaced. When you sleep at night, your bones are replaced. So I joke all the time that there's these things called, we all think we're like, let's see, the Super Bowl is Kansas this weekend. Yeah, Kansas is going to be a three-peat. Well, the first Kansas team and this Kansas team, they're not the same players. We call them the Kansas City Chiefs. But they're constantly being replaced, right? The Kansas City Chiefs of the last year are not the same year, and that's our bodies too. Our bodies, we just give ourselves a name, but our bodies are constantly changing over. So understanding the programming, and part of it's the genes, but part of it is these ion gates and other things. Understanding that at a deep level allows us to extend the number of years we have in quality, and also hopefully change disease as well. And if somebody's got diabetes, actually one of the cool things this year is that they just developed cells from a person. You can take a fat cell, tweak it with what are called Yamanaka factors, and turn it into a pluripotent cell, and then steer it to become islet cells that actually create insulin, and they reimplant that back in the body, and now they're not diabetic anymore. I mean, how amazing is that? And they're doing it through genetic manipulation, right? So once we fundamentally understand this, it will remove suffering and extend quality years. Because I don't want to live to be 90 or 100 if I'm suffering for 20 years of that, right? I want to live longer and but also be athletic as you guys. I have my knee is wearing out. I've got an artificial hip. It would be nice if I got my real hip back. So, but that's what I aspire to see. So, sorry, that was a long answer to your question, but hopefully. Other questions? I think we're right at time, but I'll be happy to stay around and talk to anybody afterwards too. So, oh, one more question there. You guys, the team decides. So the question was, how do the iGEM projects get decided? The team comes together, whatever team decide on the project. So you guys, it's just like, there are rules to the competition, but you read the rules and you come up with a proposal, and but the group of students basically pick the project. Good question. All right, well, thank you everybody for your attention. Thank you very much.